Welcome to my YouTube channel MK Mediguide Lectures, learning with artworks like drawing and calligraphy. Today, we will talk about the cholinergic receptors. Well, the neurons of our body are in contact with each other and affect our organs of our body by releasing various chemical substances called neurotransmitters. While the target site has detectors, which are called receptors, to which specific neurotransmitter binds and ultimately leads to a response. And thus, these receptors are divided based on the type of neurotransmitters to which these receptors respond. In the same way, the receptors which detect or binds to the neurotransmitter acetylcholine are called the cholinergic receptors. These receptors are also known as Cholinoceptors. We have plenty of cholinergic receptors in our nervous system and throughout our body. In all, these receptors are not the same structurally and functionally, as these receptors are not limited to a specific site in our body. And that's why, for better understanding, these are basically divided into two categories. That is, the muscarinic receptors and the nicotinic receptors, based on their different affinities for cholinomimetics. That is, the agents which mimic the action of acetylcholine when binds to these receptors. So here, let's first talk about the muscarinic receptors. Well, these receptors are called muscarinic because of their high affinity for muscarine beside acetylcholine. Muscarine is an alkaloid of some poisonous mushrooms. Muscarinic receptors are G protein coupled receptors, GPCRs for short, which is also known as metabotropic receptors or 7 transmembrane or serpentine receptors, and they are further divided based on their molecular structure, signal transduction, and ligand affinity into M1, M2, M3, M4, and M5 receptors. The first three are the major subtypes and more important clinically, and they are expressed in peripheral organs as well as in the CNS, while the M4 and M5 receptors are present mainly in the brain. Functionally, M1, M3 and M5 receptors fall into one class, while M2 and M4 fall into another class, as the odd number, that is M1, M3 and M5, are coupled with GQ protein, and once stimulated, they activate membrane-bound phospholipase C, generating inositol triphosphate or IP3 in diacylglycerol or DAG for short. IP3 causes an increase in intracellular calcium, while here diacylglycerol phosphorylates several proteins through the activation of protein kinase C enzyme, and thus it eventually leads to the generation of response. Here, in contrast, even-numbered muscarinic receptors, that is M2 and M4, are G protein coupled receptors, and once stimulated, they inhibit adenyl cyclase and thus reduce intracellular saccharine messenger CAMP and consequently decreases the intracellular calcium level, while M2 is also responsible for the activation of potassium channels. Now, let's talk about the nicotinic receptors. Well, these receptors are named nicotinic after they have high affinity for nicotine beside acetylcholine. Nicotine is an alkaloid of tobacco. Based on location in selective agonists and antagonists, two subtypes are recognized for nicotinic receptors. These are the neuromuscular nicotinic or NM and neuronal nicotinic or NN receptors. We will talk about it later in this video, but for now, all the nicotinic receptors are composed of 5 subunits that form ligand-gated ion channel, and their activation causes opening of the sodium channels and the rapid flow of these ions through these channels, resulting in depolarization and in action potential as a response by the target cell. Now, let's discuss about the locations of these cholinergic receptors one by one and how the target site responds when the receptors on it is stimulated. Moreover, most organs have more than one subtype of receptors, but usually one subtype predominates in a given tissue. So, first, let's talk about the M1 muscarinic receptors. Well, these receptors are present in nervous system as well as on some of the autonomic effectors. Like, these receptors are present in CNS where they are concerned with cognitive functions. Beside this, M1 receptors on sympathetic postganglionic neurons result in onward transmission of impulses. Furthermore, M1 receptors are also present on some free ganglionic neurons, causes inhibition of transmitter release. Besides this, M1 receptors are also found on the stomach parietal cells and upon stimulation causes the secretion of gastric acids. After this, we have the M2 receptors. Well, M2 receptors are sometimes termed as cardiac muscarinic receptors because of its presence on cardiac tissues. And in response to stimulation, the heart rate and force of contraction of the heart decreases and mediate vagal bradycardia. In addition, some M2 receptors are also present on smooth muscles in combination with M3 receptors where they cause contraction of the target smooth muscles. Furthermore, M2 receptors of some presynaptic sites lead to the inhibition of neurotransmitter release. The M3 receptors are widely found on the smooth muscles and glands of our body. The M3 receptors on the iris, circular smooth muscle of the eye causes miosis, while these M3 receptors present on the ciliary muscles causes accumulation of the lens for near vision. In these, both effects also decrease the intraocular pressure. Besides this, the M3 receptors are also found in the lungs, where the activation of these receptors causes the contraction of bronchial smooth muscles and stimulate the secretion of bronchial glands. In addition, the smooth muscles of gastrointestinal tract also have the M3 muscarinic receptors, where the smooth muscle of the walls of the GIT contract and ultimately leads to increased tone and peristalsis, while the smooth muscles of the sphincters relax. With this, the glands and secretory cells of the GI tract like salivary, gastric, pancreatic and small intestinal glands also have the M3 receptors, and once they are stimulated, it leads to increase in the secretion of these glands and secretory cells. Besides this, the cholinergic receptors in our urinary bladder is also the M3 receptors. Here, the M3 receptors on the detrusor muscles in the wall of the urinary bladder are concerned with the detrusor muscles contraction, while the M3 receptors on trigonal and sphincter muscles are concerned with the relaxation of these muscles, and these effects are related with voiding of urine. In addition to it, the M3 muscarinic receptors are present on vascular endothelial cells. All blood vessels are dilated in response to these receptors' stimulation. This vasodilation is primarily mediated through the release of an endothelium-dependent relaxing factor, ED or F for short, which is nitric oxide, which diffuses a smooth musculature, where it causes relaxation, while only few are dilated directly through these M3 receptors activation. Muscarinic receptors of the sweat glands are also the M3 receptors, which in response to stimulation increase secretion. Last but not the least, M4 and M5 receptors are largely confined to the CNS, and their functional role is not well understood. However, all muscarinic receptor subtypes are present in the central nervous system, which serve diverse functions ranging from regulation of cortical excitability, memory and learning, pain processing, and brainstem motor control. In addition to it, recently it has been discovered that cytokine secretion from lymphocytes and other cells is regulated by M1 and M3 receptors, while M2 and M4 receptors affect cell proliferation in various situations. The second type of cholinergic receptors are the nicotinic receptors. Nicotinic receptors are further divided into the NM and NN receptors, as we discussed earlier. The neuronal nicotinic or NN receptors are found in various sites like autonomic ganglia, where the stimulation of these receptors causes the activation of postganglionic sympathetic as well as parasympathetic neurons. Also, the nicotinic receptors on adrenal medullary cells are the neuronal nicotinic, which in response to stimulation leads to the release of catecholamines, that is adrenaline and noradrenaline. Furthermore, in receptors present in the spinal cord in certain areas of the brain where they may be concerned with excitation or inhibition. Lastly, we have the neuromuscular nicotinic or NM receptors to discuss here. Well, these receptors, as the name indicates, are present in neuromuscular junctions, specifically on the skeletal muscle neuromuscular end plate, where these receptors are concerned with the contraction of our skeletal muscle fibers. And with this, thanks for watching and support us by subscribing to our channel.